So welcome to PHAVES number 12. We're back, and this time we have Betty Kutter, and she needs no introduction, but she'll get one anyway. And then today is all going to be about a kind of a new format of just a casual chat, but like a little bit of a story time mixed in with question period to talk about Betty's last 60 years as a phage researcher. 60, so more than most of us can say. Her lab at Evergreen State College has been open for 49 years, the phage lab, and she has remained continuing to run that lab, even though she's a professor emeritus now. And she said to me that the only time it's ever been closed is this pandemic. So. And it's back open as we hear. So Betty is an exemplar of really a lot of what we think is important and what drives us as Phage directory, and what we're trying to do to build community is all because of people like Betty. Really, Betty herself as the inspiration, and we're trying to digitize some of Betty's spirit of collaboration in this Phage field is really kind of the heart of a lot of what we're doing. So it's very special to have Betty with us. And I met Betty in 2013 as a first year grad student studying phage, and I went to Evergreen for the first time. And Betty was really inspiring to me. She talked about not just science and things, but about balancing your life with your research in a way that I hadn't really heard at that time, and about being a woman in science, and about some of the challenges she's had to face with that. And so I think you'll hear lots today, and maybe some stories you've heard, maybe some new ones. And yeah, I will kind of pass it over to Betty now so that she can maximize her time with you. And feel free to introduce yourselves in the chat, and yeah, introduce yourselves, ask questions, and we'll kind of see how it goes for questions, whether we do them via me reading them from the chat or from you guys unmuting yourself. Just use the raise your hand function. You can find it if you do want to ask something, and we'll experiment with that too and keep this a little bit more casual. So with that, welcome Betty, and it's all yours, whatever you'd like to start with.
Okay, welcome to everybody. I think what I'll start by doing in terms of thinking about all the phage things that have happened here, and so many of you have wound up at one or many times being here for the Evergreen Phage meetings. Here I have the view out into our woods and a few of the abstract books and so forth that we've experienced over the ages. We've now had 23 Evergreen International Phage meetings here, and the plan is this coming August to have the 24th. This time, what we want to do is to have it be done online at the same time, so that the 350 last time were too many people, really, but to have a way that is open for people to do it for a very minimal cost online, and people who actually come to the meeting, the registration fee is usually just enough to cover the rooms in the dorms and so forth. So I think that's one reason it's been popular. It's probably the least expensive in general of the phage meetings. I thought I'd start by talking a little bit about my background, which some of you know, many of you know probably, but I had never thought that I would do anything biological as I was preparing to decide what I was going to go into in college. I'd spent a year in high school in Germany as an exchange student and gotten very interested in international affairs and spoke German as easily as English. It was thinking about those kinds of things. But I decided that really that what I needed to have be my primary major in college was mathematics. I had a strange reason perhaps for that, I knew that most people thought women couldn't do mathematics, and I knew that that was something that came very easily to me. My dad had sort of raised me as my son. He was an electrical engineer, and I'd done math stuff all my life. I wound up out of a class of 100 in the mathematics department at the University of Washington. 3 of us were women, and I was number 1 in the class, and one of the 2 other women was number 2, and um, the third one was only about a third of the way down. In other words, at that point in time, people were discouraged in many ways from taking mathematics unless what they wanted wasn't available for anything else. But I also, I had gotten engaged actually after my sophomore year of college. And my boyfriend was in medical school, and he talked me into taking a zoology class just so I'd better understand what he was saying. Well, it turned out that the teacher in that class, the class that I took, had gotten her bachelor's degree in mathematics as well. And um, she made it, instead of just being something that memorized all sorts of names of plants and animals and all sorts of things, and the kinds of things that I thought of biology as being, she taught it in a way of how and why, and mechanisms that were beginning to be understood of biochemistry and things like that. And um, so even though I graduated top in my class in mathematics, um, my husband and I decided that we'd go somewhere where I could study something that was somewhere in between. And where we wound up going was to the University of Rochester, New York, where he studied physics, and they had a department of radiation biology and biophysics. So I could do things that were both molecular and physical, um, and didn't know just what I'd be doing. When I first got there, it was mainly the main things they were doing had to do with treating people using radiation for treating cancer and all of those sorts of things. But then a few months after I got there, um, a person named John Weberg came to give a talk, and he was just finishing his postdoc at MIT in the lab of a man named Salvador Luria. And Salvador Luria was one of the people who wound up about 8 years later sharing the Nobel Prize, and they were in bacteriophage work. And he started talking about these amazing viruses and the things, various things about them. And the chairman of the department took me out to lunch with him and with John. And after about an hour and a half, the chairman walked away and disappeared. John and I spent another few hours together, and we decided he was going to come to there to be on the faculty. He was interviewing for a faculty position, and I was going to be his first grad student. So bacteriophage became a major love of my life, and I still, that's still been what I work with and have fun with. At that point, everything that was bacteriophage that was being done, usually most of it related to one particular bacteriophage, T4. And the Salvador Luria, who John had done this postdoc with came from Italy, and in the time just shortly before the war years, then he had been up in France, and he had started working with phage, and that became um, the major thing that he was, or a major thing anyway, that he was doing. And um, John was a biochemist, was doing molecular biology kinds of things with phage. And so my thesis was on the transition from the bacterial metabolism to the phage metabolism after T4 infection of E. coli. So it was very molecular at the same time, but you know, I think all of you have seen T4 and know what beautiful structure it is. This was something that was both biophysical and not memorizing all sorts of parts of the body and kinds of things, but something that was working in a very interesting way. To go ahead then, and the idea that this little virus could get inside a bacterial cell and make the bacterium stop doing everything it had been doing within minutes, and then take over the whole machinery of the cell and convert it into a whole group of things that allowed it within half an hour to make 200 new phages. That fascinated me. So, and still the main thing I do is work with bacteriophage T4. I'm 81 years old now, and it's still, shall we say, one of the main loves of my life, this crazy phage. So during that period of time, I went to while I was in graduate school, I went regularly to meetings that were held at Cold Spring Harbor on Long Island. Max Delbruck had started those kinds of meetings there. And when it came time then that Sig and I both graduated, things were a little more complex for me. During the course of my time in graduate school, I had added 2 sons to our family. My first son went to his first seminar when he was 10 days old and spent his first 3 months living in the lab. We had a seat for him where he could sit in there, and for the last month or so he was there, there was a jolly jumper hanging in the door between the 2 rooms of the lab. So, and I remember one time when I had left him asleep while I went to a talk and came back, and he wasn't in his bed in my boss's office, and I walked into the lab, and there he was sitting in his infant seat on the lab bench, where John was counting plaques and colonies, and the 2 of them were chatting with each other. So they didn't really realize I was there for a few minutes. And, I really realized that working with phage in particular was something that you could really do and tie in with whatever else was going on with your life without things having to be very different. So then after when we graduated and I had the 2 kids, we wound up going to the Charlottesville, Virginia, where Sig got a good spot. Sorry. And at first I was just staying home with the kids. And that was a little hard to do, much as I loved them. But after a little while, a fellow who was a biochemist and biologist who was living in Washington, D.C., but who had visited us in Virginia, came to give a talk. And I spent time with him, and I decided, nope, we'd been there half a year already. I was going to start trying to do things. And what actually happened was that I had gone to his talk and I had asked questions. I've always been a person who asks lots of questions. And afterwards, several of the people from the biochemistry and biology departments had come over to introduce themselves and to see who I was. And I wound up getting offered a job in the biology department, a postdoc position. Rolf Benziger just happened to have a postdoc position open that he was about to look for somebody for. And I was offered that position. And he was somebody also who cared a lot about little kids. So during that time, that next few years in Virginia, it was quite wonderful. After the first year, he pushed me to apply for a grant in my own name. And I thought that was kind of crazy, but I applied for it and I got a grant from NIH in my own name. And it was a 2-year grant. And then when it came time to renew it, I applied again and I again got a grant. And I decided that was about time that I should go and talk to the chairman of the department about applying for a faculty position. They had a couple of them open. And I did. However, the chairman of the department immediately got up in a great huff. The idea of a woman with young children being on the faculty was absolutely impossible. And if I had the audacity to apply for it, not only was I guaranteed not to get it, but he would no longer sign for my grant. That was the best thing he could have done for me, actually, because that was a strong enough thing for my husband and I together to decide that he would leave his very good position in the astronomy department there, and we would leave the university and we would head back to the West Coast where we preferred to be anyway. And my parents had just that year been sending us information about, they were in Seattle and there was this new college opening up here in Olympia, the Evergreen State College, and there were various writings they'd seen about it, and they sent us some information. So we just went ahead and wrote there, first of all, and it turned out that just about a month later, they were going to have, in that first year of the college, a pair of their deans coming down the East Coast interviewing various people for positions for the next year. And so we went up, this was just before Thanksgiving, and on a park bench in front of the White House, we sat being interviewed by these two deans. And within a month after that, we'd both been offered positions. The college was unusual enough that rather than it being a problem having kids, they were trying to get all sorts of different kinds of role models. And the idea of two people both on the faculty with young kids fit a niche that they hadn't filled yet. So we wound up accepting those positions and moving out in the early fall. I had come out in the spring for a visit, and I had assumed that it wouldn't be possible to still keep doing research. But when I went in to talk to the provost, who was an ecologist actually, I commented that that was the one thing that I would miss. And he said, oh, we want people to do as much hands-on as possible. See if you could possibly get your grant transferred here. so I did, and the National Science Foundation, it was NIH, that one grant, the NIH was willing to transfer it here. Our first lab building opened in January, and my lab opened the day that building opened. And we had ways then that students could come in and do work. I wasn't tied to some of the kinds of things that are expected at most major universities. Um, and, it was something that I had money that I was able to hire two of the wives of faculty to run the lab for those first 3 years. And by then, uh, I was involved enough and had enough going on that, and my kids were getting big enough that we could go on. And at that point, then I applied to the National Science Foundation and NIH for grants. And Phil Harriman at the National Science Foundation liked so much what we had been doing that I got a grant from him to keep the lab going. And in fact, he took me under his wing to the degree that um, I always had a grant from him until through the year 2000. Uh, it really helps when you have people who appreciate what you're doing. The night back after I'd been there a few years in 1978, 79, I took a year of sabbatical and went down and spent it at UC San Francisco in the lab of Bruce Alberts. And he was doing partly phage work and partly other kinds of things, and had come up to a couple of phage meetings we'd had. The um, second year I was there, I decided, you know, I really wanted, I was still working with phage, but trying to go all the way to the East Coast was expensive, and I couldn't take my students and so forth. And there were a group of people in the phage field who were coming to San Francisco for that was where the Biochemistry Society was meeting that year. And the whole group of them came up afterwards, and a few people from the University of Washington and Oregon and Seattle came, and we had a very informal phage meeting here at Evergreen, after which I took several of them backpacking up in the Olympics. And that wound up being our first phage meeting. From then on, every 2 years, uh, usually in the first week of August, we've had phage meetings at Evergreen. But those first meetings, in fact, the first 10 of them were called T4 meetings. All of the phage work, almost all of the phage work going on was with bacteriophage T4, which and the people working with temperate phages went to Cold Spring Harbor, and a few of them, of the T4 people, also went there, but they all started coming to the meetings here at Evergreen, and we'd do hiking afterwards that some of them would come along. It's a really nice informal place for meetings. And we've now had 23 of them. The 24th biennial meeting will be this next summer. Hey, Guy, hello. Guy Plunkett is one of the people who's been around from those early days doing phage, and his name just popped up. And I recognize a lot of you, and including people from India where I gave a talk last a year in not last December, but the year before. So phage has been my life and my hobby. And we've had as many for those early phage meetings, there were usually anywhere from 30 to or 40, and then it got to be 100, and we'd do some hiking in the Olympics afterwards with some of the people, or and take everybody up for the last day of the meeting by bus to Mount Rainier to unwind and have some pleasant times with each other. So that, I think, has had a significant role in keeping the phage community as a community. And at least this summer again, I am planning to have, did I mention that already? To have a meeting here that we do with as many people being able to come online as they want to, or to be able to come in person. And that's already set up at the end of the last meeting. I thought at the last meeting that was probably going to be the last one, and with 350 people there for that, at the end I said we'll do at least one more, maybe even two more. So the question, what I wanted to do is then not only give my history today, but to spend a little time talking about the early years of phage and the development of phage. And the way it's been involved in much of the development of molecular biology. As I think I mentioned, John Weberg had just been at MIT in the lab of one of the three people who wound up getting the Nobel Prize for phage work. And he, when he set up the lab there, made it in a way that it was easy for me to keep doing it with having kids and so forth. In fact, shortly after he'd gotten there and the lab was all set up, I had been off to a class, and I had left my then 3-month-old asleep in his infant seat. And when I walked into my office, he wasn't there. And I went into the lab, and there he was sitting in his infant seat on the lab bench with John talking to him. And one of the nice things about having a lab here at a place like Evergreen, it's been easily possible to do it so things are informal, so that students can be in and out of the lab, so that they can easily be moms and dads and so forth, and going to classes a substantial part of the time, but also phage is such a wonderful system. You know, if it comes a time when there's a few months when you can't work with them, you just stuck them in the refrigerator. They're waiting for you when you get back. And phage went on to become such an important part of the development of molecular biology.
Betty, I wonder if, can you talk about the part where it transitioned from molecular biology to when you started talking about like phage therapy and you went to Tbilisi, like that period of transition?
Oh, I'll be glad to, yes. Yeah, initially it was all molecular biology. And then what happened was that I was when we were working on the sequence of bacteriophage T4, actually my lab wound up leading the work to doing the sequencing, but we had collaborators, including a wonderful fellow Fumio Arisaka from Japan, and a wonderful fellow from Moscow, and a fellow from Germany. And tied in with that, I wound up then at one point being able to get a grant that let me spend 4 months in the Soviet Union. And this was in 1990. And during the time I was there, first of all, there were a lot of people who'd been working with Phage for a long time. You know, during all the years when it hadn't been done so much in this country, both the Republic of Georgia and in Moscow had been doing a lot of really incredible work with Phage. So I went for 4 months at the beginning of 1990. And during that time, I wound up going twice down to Charlottesville, Charlottesville, what am I saying? Um, Down to the Republic of Georgia and working with people there, meeting wonderful people, and was able, with the help of my friends at National Science Foundation, to bring a couple of different people over to work in my lab. And I wound up going back to spend some time there every couple of years. And at that point, we discovered, not only was phage the basis of molecular biology, but they were actually treating people. And that it was something that they, in fact, had been doing ever since the 1940s. And so that was something that I started getting interested in, and wound up being able to work back and forth with people there, and to begin to do some work here with phage. I had always been interested in complementary medicine, and in the early years I was here, one of the people who taught with me was a naturopathic physician here who was just about to help open the second naturopathic college in this country up in Seattle, and I was on their board for about the first 40 years of the college there. And there was the idea of, gee, phage can be used therapeutically. And one of my students that first year that I was teaching here who had gone on to become a naturopath in Portland, was very excited about using phage, and got permission from their board. And so we brought phage from Georgia, and she's close to retirement age now as a doctor of Oriental medicine and naturopath, and still using phage. So at that point, that led to a phage therapy becoming part of our phage meetings here. Instead of being just T4 meetings, they became now the Evergreen International Phage meetings. And we had people who were coming from all sorts of different countries, and we had sessions on basic molecular biology and sessions on phage therapy. And it became something that was sort of taken for granted as something that was possible. And there were several other naturopaths here who began using phage. And then about a decade ago, one of my other students from that first year, who I'd lost touch with, turned out had become a podiatrist, doctor who worked with feet. and they were opening a new clinic here in Olympia at one of the hospitals that he was interested in working with. He was up in Tacoma. As I say, I'd lost touch with him after that first couple of years, but one of my neighbors went to this meeting talking about the opening of the clinic, and he asked, are they going to let them use phage there? And so this former student of mine went up to him afterwards and said, how do you know about phage? And put him back in touch with me. And he's now on our board of our Phage Biotics Research Foundation, and spent several years of using phage occasionally for particularly problems with the front part of feet, and found that it was working very well. At that point, his main clinic was over near the ocean. Now he's getting involved in a clinic here, and he's going back to wanting to use phage more often again. And pretty soon there's going to be more phage work actually happening here. But particularly, I don't think he ever got any formal permission back initially to use phage. It worked, so he used it. And long before other people were starting to use phage in this country, then he was having great fun using it. And turned out later then that a couple of people who were supporters of some of our research work at NSF and all learned about what he was doing and said, and he wound up getting formal permission and so forth. But that's how phage therapy, to a significant degree, started here between him and the naturopath in Portland. And now it's gradually coming to be much more. I'd like to know more about some of the people who are here and some of the kinds of questions you have, and get into some discussion.
Perfect. Yeah. And I'm looking at the chat and people are saying really nice things. Like Natalia Portilla says, 5 years ago in my first year at college, I was inspired by your contagious phage love. Thanks for helping a new generation of phage scientists get inspired. I'm paraphrasing a little at the end. Oh, yeah. So anyway, we're definitely going to be sharing all these comments with you at the end, Betty. So if we don't get to reading them all, feel free, everyone, to write whatever you like. Jean-Paul Pernay says, hi, Betty, nice to see you. You witnessed phage therapy in Georgia. Now the new phage therapy initiatives are all around the world, like in the US, following current drug development models. What would be your advice to them? There's a question, our first question.
Well, my first suggestion would be to find a good small college somewhere where rather than it being primarily research oriented, they are interested in people who want to learn to work in labs and are interested in eventually going to medical school and things like that. There are a couple of other colleges that I think that are doing that now, and teach there and open a phage lab. And phage are, they're safe. They're such wonderful ways of being able to work with them and ask all sorts of basic questions. A lot of the understanding of molecular biology developed through work with phages, where you're not trying to work with something so complex as animals or bacteria or things like that, where it's something that there are lots of different kinds of phages. They're doing different things to the bacteria. You can you know, almost all the work in my lab has been done by undergraduates um, who wind up going on then often to do medical kinds of things in graduate school. But um, one advantage of being at a college is it's often more flexible in the kinds of things people do, and you don't have um, a big, you don't have a research um, set of people in charge who are making all kinds of rules. That may sound strange, but excuse me. I thought I was over my cough that made us put it off for a week, but it's back again a bit.
Shall we go to another question?
Yeah.
You can take a breather from Shauna. We'll call on you, Shauna. See your hand.
Hi, Shauna.
Hi, Betty. I I don't think I can turn on my video, Jess.
There, go ahead. I have
All right.
allowed.
It's always nice to sometimes see people's faces there.
Greetings, Shauna. It's really good to see you.
You too. I know we've been meaning to catch up for a while. I was wondering though, so we do phage selection, phage isolation for phages we want to use for therapeutic purposes later. And we always try to select phages that have the broadest host range. But then I'm finding we keep coming back and isolating really similar phages. And I was wondering from your perspective, like with all of your molecular knowledge and your mathematical background, if you think there's some type of test or some type of characteristic we should be looking for when we're trying to isolate phages that maybe we're missing in our standard phage isolation pipelines right now.
Well, one of the things that we've done, and particularly I did it in a large way a little over a decade ago, the last time I had a full class, we had everybody in the class isolate phages. Most of them did it from our local sewage systems. A few went out to farms and things like that. Got a different phages. These were phages that I'm talking about here. And then we used the ECOR collection, the E. coli collection of reference, and we tested all of those phages against that whole collection. And we found that they had a whole range of different patterns of which ones they would hit there. And that gave us a whole family of phages that had, and a number of them were T4 related, but some were different than that, but they had different patterns of what they were infecting. And so then, so now we have a set that if we're looking for things, we get, they're related, many of them to T4 or to T5, but they're different. And you can do the same sort of thing with Pseudomonas. There are a whole lot of different strains of Pseudomonas. That's the other main kind of bacteria we've worked with through the years. And again, if you take a lot of the different Pseudomonas strains and test your Pseudomonas phages against them, then you can get ones that you know are different. And then you can use those to test on a plate against bacteria from a patient, for example. And you may want ones that are broad, but it's sometimes useful, as you say, to use ones that have a very broad host range, but actually to use ones that particularly work against, and it's so easy to test on a plate when you're talking about things like E. coli and Pseudomonas. The other one that we've worked with a fair bit is staph phage, the one gram positive. there's not nearly the same kind of ranges of things that we found with that. But again, we find ones that seem to we can do a lot of work with understanding them better, and we can have the students do a lot of things like that. And then that can set up a real research program at a small college that costs almost nothing. That I mean, I've had groups of 7th and 8th graders in there working in the lab sometimes. And you know, it doesn't take expensive things. And it really, that's been my joy. And I found being at a small college myself and being much of the time the only basic research lab there, I have an awful lot of freedoms. And they've let me still keep my lab there, even though I retired a decade ago, and I still have a few people working in the lab. And a number of them have gone on in various ways. One of them is working there now in Brussels at the hospital and at the major hospital. Others are working in other major places and still doing things that build from their phage. Does that make sense?
It does. Thank you, Betty.
And, you know, I have even had grade school kids in there and let them show that they can do a bit of some things. And when you catch people early on, feeling that doing that science is something that people do, something that people can do, and they develop a love of exploration at a point where they're new in the world. It's been, but it's not only that. Right now, my lab recently has been opening back up, and it's being sort of led by 2 people who had graduated about a year ago as everything was shutting down, and they're helping do that. But I also have 2 current students there who are, one is a woman in her late 30s and the other is in her early 40s. And they're people who have come back to college. One of them was a very close friend of a fellow who was a student of mine a decade ago, who's now down in the Bay Area. And, you know, it's really nice having people then be able to do things coming from a lot of kinds of backgrounds. One of the things we're doing now is really starting to test a whole group of different phage infections anaerobically.
Yeah, this would be really interesting.
Yeah, and anaerobic work hasn't been done very much at all. And we had, just before everything got into lockdown, we had gotten into setting up a good small anaerobic chamber and done a little bit of it. And actually, the first time we had done some work anaerobically was back when we were working with phage against some bacteria from cattle. And it was clear that if you're thinking about using phage therapeutically, a large part of where will be microaerobic, not, you know, it doesn't need to be absolutely anaerobic. That's not what it would usually be. But if you're talking about something in many parts of the human body, they're relatively microaerobic. So think about some of those kinds of things, or isolating phage from the local streams and things like that.
I'll definitely think about doing the anaerobic isolation, or at least testing on our collection. So thank you.
Awesome.
So yeah, I think you have, Shauna, you have a new place where you are, right?
Yeah, I'm in Zurich, but I'll try to catch you this week and I can tell you how I ended up here.
Anyway, I'm looking forward to talking with you about what you're doing and, getting people, even people who will never necessarily go on to be phage scientists or maybe even never scientists at all, but having them feel comfortable with the idea of phages and with the idea of using phages therapeutically. And now at UC San Diego, they wound up discovering that phages could work very well when nothing else did sometimes. The stories are all around. But I think, to me, my most important use of phages has been to be able to help people get excited about science. And I should mention, by the way, that these two women who have just, one last spring and one now get started working in my lab, they each have teenage kids. And I've told them as we're getting going that they're welcome to occasionally bring their kids into the lab to help them.
Always good to get them started early.
Yeah. And both of them have kids who are, one is a senior in high school. The other is only 14, but excited about science and having people who have never had a chance to do some of the things they would like to until they're in their 30s and 40s, and being able then to have that be a family thing is something that's great fun.
Betty, I want to get to a couple more questions. We have one from Pranav and Apurva who have their hand up. So we're going to swap Shauna out of the spotlight and bring in Pranav. And before we get to the question, just want to mention, Betty, that if you're seeing the chat, there's people saying they're coming here from Mexico, Peru, Tajikistan, Ukraine, Germany, UK, India.
It's not Deutschland.
So just in case you're wondering where people are from, it's everywhere. That's the answer. So Pranav, Apurva, would you like to ask your question?
Sure. Betty, it's again a pleasure to hear you recount your entire story, your personal story. It's always so amazing to hear the entire experience and your entire history with the world of phages. I want to ask you that what do you think is the most effective way to talk to doctors about the power of phage therapy specifically? Because a lot of them know conceptually or theoretically about phage, but they don't know that this can actually be applied to their own practice, to their own patients, to treat antibiotic-resistant infections. So what do you think is like the most ideal way to communicate to doctors and medical professionals about phage therapy?
Well, one of the things is to start with giving talks at medical schools. And here in the States, there are the regular medical schools, and there are two major naturopathic schools, one in Seattle and one in Portland. And years ago, I used to every now and then give talks. Haven't done it in recent years, but it's easy to get invited to give a talk about phage in the medical school, and the students have lots of questions, and then they grow up thinking that it's something that is something that's an option and ready to do that. One of the problems is getting the permission to be able to use phage, and one of the things that at our last phage meeting here, well, one thing is that there are people from NIH who come to our phage meetings. We've done now, as I said, 23 biennial phage meetings. And among the 350 people who were there last year, and even ones who are coming significantly earlier, there have been several different people who are associated in one way or another with NIH. And in a way that then the people who make the rules are more likely to be able to listen, to be able to get permission. Jean-Paul, can you talk a little bit about ways to get people willing to go to medical school? Can you add in some questions about that, Jean-Paul Pernet?
I think Jean-Paul just popped out and back in, so I'm not sure if you can hear us, Jean-Paul.
I was kind of kicked out of the meeting. I don't know why.
Sorry,
I didn't hear a question.
Betty, can you repeat? Yeah.
I was just talking to you they were asking about how to get people interested and willing to try to use it therapeutically, even if they're learning about phages. And I was saying that one of the things that I've had some ties at the medical schools in Portland and in Seattle, and haven't done it for a while, but periodically I have given talks to their students about phage and about phage therapy to the students in the first year class, usually. And I think that getting people so that they're aware of that at that stage in medical school is a good way. What about you? I mean, you certainly are there in
Well, I felt that it was not that difficult to convince medical doctors in using phages. They really want to do this, but of course they have been some kind of brainwashed to do this according to the current rules, the current regulatory framework, and that makes them a bit afraid. So they are convinced that phages are helpful, but they are a bit afraid of some consequences when things should go wrong. But in my experience, it was not that difficult. It was easier than I thought, at least in the military hospital.
Yeah, can I jump in? So my name is Gerard. I am a quality control at one of the
I can't quite hear.
Oh, sorry. Can you hear me guys now?
Now, yes.
Yeah.
So my name is Gerard. I am one of the quality lead in one of the top phage companies here in United States. The name is Adoptive Phage Therapeutics. So what we have done, we showed the doctor that the phage has no receptors on mammalian cells, so they are not dangerous to use. They have the receptors only with the bacteria. This is one thing, and we already helped them. We already showed them that there is also a benefit of doctors in this therapy too. So that's why now we are working with Hopkins. We are working with a lot of prestigious universities, schools, and medical doctors, and a lot of other stuff by showing them that they are very safe. And we are only one of them. We are using IV phage, we are using IM phage, and we are using a lot of different type of phages. So if anybody need any kind of help, and I can see there is a question by Mr. Tom from Ireland, and he has a very good question saying hello from the UK. Great to hear you talk again. What do you think the phage therapy will look like in the future? It's an organization, regulation? Yes, right now there are a lot of people are coming into the phage therapy. We have a lot of IND, it means investigative new drug application with FDA. So FDA has already developed a different segment, which is in the CBER. If you want to add, or if you want to start any phage therapy from any standpoint of view, like prosthetic joint infection, if you want to start UTI trials, if you want to do cystic fibrosis, if you want to do like some kind of retinal infection you want to treat, or any kind of multi-drug resistance. So right now, the FDA knows how to deal this. So there are a lot of regulations, a lot of other things. And the only problem was how to produce the phage in the bulk. how to get rid of the endotoxin, how to get rid of the genomic DNA, how to get rid of the host cell proteins. These are the real questions. I think you should emphasize on that. So I hope answer the question. If you have any question, I will write my personal email ID. If anybody wants to ask any question, I hope I will, if I have answer, I will help. So thank you.
Hope for chiming in.
Yeah, so, and it ties into with the kinds of regulatory groups. As I said, the naturopathic physicians, both in Oregon and Washington, had a very easy time of convincing their boards that it was a useful thing and a possible thing to do. Whereas I think it's taken longer for the MDs, but there are things happening now at the national level that it seems to be possible, and particularly starting to use them for wound therapy and things. After I gave that keynote talk last December in India a year ago, I went up to the meeting at the military hospital in Berlin, and there were people from every European country there, a group of about 60 people at a meeting talking about using them routinely for wound treatment in the military there. And it was a very interesting conference. And it seemed like part of it was developing particular kinds of applications and developing good procedures that were shown and places that you could get them and ways they were purified and things like that. But it was a 2-day meeting of people. And even though that was the first time that they were trying to get approved to use there in Berlin, it was clear that they'd treated already a few dozen people, and that most of these people from other countries were occasionally having doctors that were in a situation where that was something that, wasn't being marketed specifically for that purpose, making it easy to get, but things where people could get their hands on them from people who were researchers and knew that they could go ahead and use them.
Just one quick thing. So we're a little past the top of the hour, and I want to make sure everybody knows if they have to get going, you're welcome to just head away, leave if you want. I don't want to make you feel like you have to, that you're not sure what time it ends. We're going to keep it open till 1:30 Eastern, so 25 more minutes, and answer some more questions if that's still good with you, Betty. But everyone's
It's definitely okay with me, and I would love it if there are more of you who are from places like Pranav, where you are occasionally using phage, and what you have found as ways to help get permission, at least on an individual level, even if they're not being sold in the pharmacies. I mean, I think there's that 2 levels, and it seems like there are quite a few people here. By the way, Naomi Hoyle is now back in Washington, who's been in Tbilisi for the last more than a decade, and she's just been going through everything to be able to practice again, actually, in this country. She was my student who went on to have a major role working with, did medical school in Tbilisi and did major work at the Eliava, and is still formally also on their staff to some degree, even though she's here. I should have called to tell her that this was happening today, and I didn't think to, but anyway, you're going to have it up online, I think, right?
Yeah. I want to go to a question from Jessica Neubauer. She's been holding up her hand for a little bit, figuratively, so and then we can go back.
I see the hand. That's cool.
Okay, go ahead, Jessica.
Hi, hello from Vienna. I'm yeah, I'm a big fan of phages, obviously, and your work as well. And I remember in my undergrad, my first microbiology lecture was 2 slides, not even 5 minutes. Our professor said, "Oh yeah, and then there's phages, and they used to use them as antibiotics. And now on to the next topic." And I remember sitting there going, "Wait, what?" And I don't remember anything else that he talked about that day, but just those 2 minutes. And yeah, since then I'm hooked as well. And like I said, I'm just starting out. I'm a first year grad student now. And do you have any tips for us small baby scientists?
We can't
Well-
see your face, Betty, just a sec. Can you put your video a little bit down? We can see your forehead.
Right. There. I think one of the things is to become friends with doctors who have fairly broad practices, and particularly who treat wounds. That's the place where it seems to be most often that people start being willing to use phage, as opposed to things that are internal. And find both, but find a practitioner or 2 or 3. You may get to know them through friends. You may get to know them through something you put online. So long, Michael. Mike Skurnik is just leaving. I'd love to talk to sometime in person. But phage, and one of the main things is to get it so that they can be prepared in such a way that they know that it's sterile, though at the same time not so sterile as to kill the phage. I found that people get really excited about them when they hear about them, and I think one of the main things that a few of you are being able to do, like you in India, Pranav, and the people there in Belgium and so forth, to get information out. In this country, in the places where they've been allowed to be used, it's generally seems to be as if it were the last ditch kind of thing. And I think that the most important thing is to help work with doctors to get to the point where it's the early part of the treatment, where it's one of the original things that one thinks about, one of the natural things, rather than waiting till somebody's on death's door and hoping that you can get enough phage to them in enough time to work. It depends partly on what you're treating. And also, I think often for fairly simple wound treatment is a way that's external, that you can, find people who are doing some of that, find ways, make phage and get them approved that are in a sterile enough way of a few of the broad phages for really common external applications, and get people used to the idea of thinking about phage.
That's great.
And UC San Diego has been doing a fair bit of that kind of thing. And Carolyn there in Texas has been the middle person of preparing them for so many people.
San Diego and Texas are kind of my dream, maybe for a PhD in a few years. Ideas.
Yeah, I mean, it's really not so much the head of the lab, but she's been there in the lab for the last 5 years or so. I'm hoping that she's going to come up here for at least a few weeks with us, as she's deciding what to do. She's just passed her PhD exam.
I think she's here, Adriana. There she is.
Adriana.
People come out of the woodwork if you mention their names.
I'm right here. Thank you.
Introduce yourself,
I was trying to be
Adriana,
in the
a little more. Can you tell people a little more about yourself and about using the phage?
Hi, everyone. For those who I haven't met, it's awesome to see you here. My name is Adriana Hernandez Morales Long. I'm Colombian, and I came to do my PhD at A&M, and I ended up producing one of the cocktails that was used for the Tom Patterson intervention that was very famous, and Stephanie Strathy, that ended up in the book.
Would you mind telling people what kind of phage it was? I don't know whether that's public
we
yet or
ended up
not.
talking with Betty for a long time about this, and it turns out that all of the phages in the original cocktail are T4-like phages that infect Acinetobacter, and it's that specific strain of Acinetobacter. And so we've been talking because it's T4 and they're all T4-like phages, and yeah, it's been a very interesting journey. And now I'm waiting for my papers, so I am twiddling my thumbs a little, not a lot, a little.
And I just learned recently from Adriana that they were T4-like, and I have not told anybody except my own lab people, because I thought it was something that was yours to share. But the idea, I worked with T4 since 1963. My lab at Evergreen were the ones who did the sequencing of T4 and some of the T4-like phages. T4 has been my my baby, my pet all my life, and I've worked with it so much. And when I learned recently that was and a real T4, not even T2 or T6, that was like bringing a major many years of my life to a lovely closure. So I've been careful not to say what it was to anybody, because it's your right to share it. But thank you for doing that here.
Thank you.
So
I had a question that was mostly so I think that your journey coming from a larger college to a smaller college and bringing a full R01 and then having so many years of funding in NSF is very unusual. And I don't know if this is the case for a lot of people that work in small colleges, but how do you see funding for research like yours and like other people's research who are interested in doing phage and phage therapy? How do you see it happening in small colleges in terms of funding? Like, do you see that as a possibility, and where would you get that funding, and how, like?
Well, for one thing, in the small colleges, where you're doing it with as part of the regular labs that students are doing, the work with a lot of the work with phages that we did was something that was part of what I was teaching. So in that sense, the college was funding the teaching. And the other thing is that there are grants available from National Science Foundation in particular, was what did it most of the time, that are specifically for small colleges. And phages can be a very good way of attracting some of that funding. When you're talking about something where you're not having to pay most of the people doing the work, that they're delighted to be able to have the opportunity to do that. And as a faculty person, you're entitled to have a lab, and phages can be done very inexpensively. So, I mean, many of the years, I didn't have more than $1,000 or so later on. But I think also there are now some people who are becoming aware enough of it, and pay attention as you work with people at NSF or at NIH. I mean, I was lucky that I found Phil Harriman at NSF, who had this program for small colleges in general of research funding that became quickly convinced that Phage were a wonderful way to spend that. And there's several other small colleges that have done some of that sort of thing, particularly up in New England. Phage are something that are safe, that you can stack away in the refrigerator for 6 months, that you can keep going with. Do any of this, sorry, did any of the rest of you have particular ideas about that?
I mean, I know Toby's been done. I think Toby's around here. I know Toby's been doing a great job in like a type of technology transfer with Phages with her nonprofit, which I think is absolutely great, with the idea of getting Phage and Phage knowledge outside of the US and then developed countries. Maybe that could, so setting up a small like research, but I think that the main issue that I've looked at, and especially coming from Colombia and knowing what the situation in terms of research is in developing countries, is usually sequencing, where everything falls short. The access to electron microscopes and sequencing is not something that is very common outside of most countries in Europe or the US. And so
that's something we didn't have access to in any of those years at Evergreen either. I found people who were willing to do it. And one of the things is, I mean, I have a small foundation, a Phage Biotics Research Foundation, that would be glad to raise money to help with the small amounts it takes for doing some of that. Or, maybe if you can find people are willing and interested to do sequencing of particular strains. I think, and in terms there in Africa, particularly in Kenya and Uganda, I know there are people now there who are doing work with Phage that sort of got nudged in that direction when I was over visiting my granddaughter in the Peace Corps. And so partly I think it's making partnerships.
Does anybody else want to comment on the question about sources of funding for Phage Labs? If you do, raise your hand with the hand raising option. And in the meantime, I want to go to Newton, who has had his hand up for a question, but we can come back to this funding question after that. So Newton, do you want to go? I will allow you to unmute yourself. Floor is yours. I see you've unmuted.
Okay. Hello. You couldn't be able to see me because the host has to allow me to.
Oh, sorry. Sorry about that.
Switch on my.
There you go.
Yes. Oh. Yes. So I think perhaps I have to talk on behalf of African people. Surely. Yes. I'm talking to you, Professor Betty from Africa, basically, and I'm Newton. I'm normally from Benin Republic, but currently I am in Kenya as a PhD student. So yes, I have really heard about you. People have been talking about Professor Betty Kutter. You train a lot of people at where I am currently doing where I am doing currently my Phage lab research. It is in the Institute of Primate Research, IPR. Perhaps you, yes, they talk about what you have done for them and the passion that you have given to them to continue on working on phages. And we believe that your legacy will not be in vain. Yes, that's what I have to say.
Thank you. They're the ones who started calling me the Queen of Phage.
That's one of the words, the Queen of Phage, the grandmother of Phage. I love them all.
Well, Phage, Phage has been my life for 60 years, so.
Okay, so we have a few more minutes. So I'm going to find a couple questions from the chat. Oh, actually, I wanted to read this one. Just a bit of a comment from Gulshan Shakiri. Very nice to see you, Professor Kutter. One of your articles rescued my project while I was alone struggling to isolate E. coli phages. However, I could not yet finish the project. Regards from Iran. So that's what the message is. If anyone else wants to ask a question, go ahead and put your hand up or type it down in the chat. Oh yeah, we have the First Lady of Phage. That's the one I was missing. Thank you, Pranav. That's another name for you, Betty, as you know.
Well, by the way, we do plan, let me take the time to tell all of you and to spread the word. The first week of August, we do plan to again have a Phage meeting here. And this time what we plan to do is not only to have a meeting here, but not as big as last time. We'll keep it down to 100 or 150, but to make it also available online, all of the talks, to make them available live online and then also able to be cut into online. And I think we're going to maybe charge something like $50, and if that's too much, then nothing. That people can have all the access they want to have. And we do still have some of the talks in fact, we have the whole set of talks, at least from last year, that I'm going to try to work with people when I can get back over to school to see if we can get, maybe we can work with you, Jessica, to get those up online from earlier years.
Excited to help make sure all those videos get shared and that we have a place for them people can come back to and find them, and to help with the online component, hopefully, of Evergreen and help make it happen. Super excited about that. I wanted to, there's a question about temperate phages, and I know, Betty, you've been talking to me about temperate phages in our lead up to this. So I wanted to maybe ask this one. Maybe this will be one of our last questions. It's from Francisco Ricardo in Mexico. Francisco Rodriguez. Says, I'm new to this amazing field of phages and I recently isolated temperate phages against P. aeruginosa. What do you think are the main challenges of working with this kind of phages?
Well, I think that temperate phages are less likely to be useful to kill, to use therapeutically. I mean, I haven't generally figured out ways. I've sometimes been able to take lytic derivatives of a temperate phage, but the temperate phages are helping the bacteria survive in particular ways rather than killing them. So I guess there might be phages that were temperate in a few kinds of bacteria, but not able to become prophages in others. But I think that lytic phages are more the way to go. I've done almost nothing with temperate phages, but I've done work with a lot of lytic phages in about 50 different countries.
Awesome.
What a life I've had.
Yeah. Well, thank you all for coming, and
everybody save the first week of August, save time the first week of August. We'll all be online,
yeah, we'll get
but
the
also,
um,
and if you have any of you who have done phage work that you'd like to either make a poster that we could post about, or perhaps be considered for giving a talk, I would love to hear from you, and we'll do everything we can to make it possible to do it electronically if not managing to come here.
do you want me to share your email address in the chat?
Yes,
Betty?
kutterb@evergreen.edu.
Okay. And so they can email you if they're interested in giving a talk or a poster, right?
And all the posters we can put up online, as well as, we will have some people here if we can. And if you're in a position, the registration fee is basically mainly just enough to cover the room and board and all that stuff. The regular fee is about $400, and for students, we make it less. And for people who don't have money, we often just take care of it. So don't hesitate to get in touch.
Yeah, Evergreen is highly worth attending.
I'll show you again
Oh yeah.
Can see it, the collection I spread out of some of the phage things through the years. You can imagine these are just a fraction of what I have at home. Say nothing of can you see them?
Yep, we can. Abstract books, all kinds of, yep, the tote bag from Evergreen. Probably, how many years of abstract books do you have by now? All of them, I guess.
Well, the first two meetings we didn't have abstract books for, but I have them for about 20 of the meetings.
Incredible. Well, thank you so much, Betty. We'll make sure you get all the comments people have shared, because there's lots of little stories about how you've inspired people. So I'm going to make sure you have those to keep.
Well, and I like to encourage all of you, and if people feel like, share my email, kutterb@evergreen.edu. And these days anybody I hear from, I'm likely to be able to answer.
Wonderful. Well, with that, we're going to say goodbye.
Phage help one to still be healthy. I mean, I'm 81, and I still several times a week walk 2 to 5 miles, and I still have a plan to sometime again come back to Tbilisi at least.
Wonderful. Well, thank you. And thanks everyone for coming. See you soon.
Bye-bye.